Class 11, Chapter 16 Excretory Products and Their Elimination. Animals accumulate ammonia, urea, uric acid, carbon dioxide, water, and ions like sodium, potassium, chlorine, phosphate, sulfate, etc., either by metabolic activities or by other means like excess ingestion. These substances have to be removed totally or partially. In this chapter, you will learn the mechanism of elimination of this substance with a special emphasis on common nitrogenous waste. Ammonia, urea, uric acid are the major forms of the nitrogenous waste excreted by animals. Ammonia is the most toxic form and requires large amount of water for its elimination, whereas uric acid, being the least toxic, can be removed with a minimum loss of water. The process of excreting ammonia is ammonotalism. Many bony fishes, aquatic amphibians, and aquatic insects are ammonotelic in nature. Ammonia, as it is readily soluble, is generally excreted by diffusion across body surface or through gill surface in fish as ammonium ions. Kidneys do not play any significant role in its removal. Terrestrial adaptation necessitates the production of lesser toxic nitrogenous waste like urea and uric acid for conservation of water. Mammals, many terrestrial amphibians and marine fishes mainly excrete urea and are called ureotelic animals. Ammonia produced by the metabolism is converted into urea in the liver of these animals, released into the blood, which is filtered and excreted out by the kidney. Some amounts of urea may be retained in the kidney matrix of some of these animals to maintain a desired osmolarity. Reptiles, birds, land snails, and insects excrete nitrogenous waste as uric acid in the form of pellet or paste with a minimum loss of water and are called uricotelic animals. A survey of animal kingdom presents a variety of excretory structures in most of the invertebrates. These structures are simple tubular forms, whereas vertebrates have complex tubular organs called kidney. Some of these structures are mentioned here. Protonephridia or flame cells are the excretory structures in platyhelminthes, flatworms, example planaria. Rotifers, some annelids and cephalochordate amphioxus. Protonephridia are primarily concerned with ionic and fluid volume regulation, that is osmoregulation. Nephridia are the tubular excretory structures of earthworms and other annelids. Nephridia help to remove nitrogenous waste and maintain a fluid or ionic balance. Malpighian tubules are the excretory structures of the most of the insect, including cockroaches. Malpighian tubules help in the removal of nitrogenous waste and osmoregulation. Antennal gland or green gland perform the excretory function in the crustaceans like prawns. Human excretory system. In human, the excretory system consists of a pair of kidney, one pair of ureter, and a urinary bladder and a urethra. Kidney are reddish brown bean-shaped structures situated below the level of the last thoracic and third lumbar vertebra, close to the dorsal inner wall of the abdominal cavity. Each kidney of an adult human measures 10 to 12 cm in length, 5 to 7 cm in width, 2 to 3 cm in thickness, and an average weight of 120 to 170 gram. Towards the center of the inner concave surface of the kidney is a notch called hilum, through which ureter blood vessels and nerves enter. Inner to the hilum is a broad funnel-shaped space called the renal pelvis with projection called calyces. The outer layer of kidney is a tough capsule. Inside the kidney there are two zones, an outer cortex and inner medulla. The medulla is divided into a few conical masses, medullary pyramids, projecting it into the calyces, singular calyx. The cortex extends in between the medullary pyramids as a renal column called columns of Bertini. Each kidney has nearly 1 million complex tubular structures called nephrons, which are functional units. Each nephron has two parts, the glomerulus and renal tubule. Glomerulus is a tuft capillaries formed by the afferent arteriole, a fine branch of renal arteries. Blood from the glomerulus is carried out by afferent arteriole. Efferent arteriole. The renal tubule begins with a double-walled cup-like structures called Bowman's capsule, which enclose the glomerulus. Glomerulus, along with Bowman's capsule, is called Malpighian body or renal corpuscles. The tubule continues further to form a highly coiled network, proximal convoluted tubule, any PCT. A heparin-shaped Henle's loop is the next part of the tubule, which has a descending and ascending limb. The ascending limb as another highly coiled tubular region called distal convoluted tubule DCT. The DCT of many nephron opens into a straight tube called collecting duct, many of which converges and open into the renal pelvis through medullary pyramids in the calyces. The Malpighian corpuscles PCT, DCT of the nephron are situated in the cortical region of the kidney, whereas the loop of Henle dips into the medulla. In majority of nephrons, the loops of Henle is too short and extends only very little into the medulla. Such nephrons are called cortical nephrons. In some of these nephrons, the loop of Henle is very long and runs deep into the medulla. These nephrons are called juxtamedullary nephrons. The efferent arteriole emerging from the glomerulus forms a fine capillary network around the renal tubule called peritubular capillaries. A minute vessels of this network runs parallel to the Henle's loop, forming a U-shaped vasa recta. Vasa recta is absent or highly reduced in the cortical nephron. Now, urine formation. Urine formation involves three main processes, namely glomerular filtration, reabsorption, and secretion that takes place in different parts of the nephron. The first step in urine formation is the filtration of blood, which is carried out by the glomerulus and is called glomerular filtration. On an average, 1100 to 1200 ml of blood is filtered by the kidney per minute, which is constitute roughly 1/5 of the blood pumped out by each ventricle of the heart in a minute. The glomerular capillary blood pressure causes filtration of blood through three layers, that is endothelium of glomerular blood vessels, the epithelium, Bowman's capsule, and the basement membrane between these two layers. The epithelial cells of the Bowman's capsule, called podocytes, are arranged in an intricate manner so as to leave some minute space called filtration slits or slit pores. Blood is filtered so finely through these membranes that almost all the constituents of the plasma except the proteins pass on to the lumen of the Bowman's capsule. Therefore, it is considered as a process of ultrafiltration. The amount of filtrate formed by the kidney per minute is called glomerular filtration rate, GFR. GFR in a healthy individual is approximately 125 ml per minute, that is 180 liter per day. The kidney have built a mechanism for the regulation of glomerular filtration rate. One such efficient mechanism is carried out by juxtaglomerular apparatus, JGA. JGA is a special sensitive regions formed by the cellular modification in the distal convoluted tubule, DCT, and afferent arteriole at the location of their contact. A fall in GFR can activate the JG cells to release renin, which can stimulate the glomerular blood flow and thereby the GFR back to normal. A comparison of the volume of the filtrate formed per day with the data of the urine release suggests that nearly 99% of the filtrate has to be reabsorbed by the renal tubules. This process is called reabsorption. The tubular epithelial cells in the different segments of the nephron perform this either by active or passive mechanism. For example, substances like glucose, amino acid, sodium, etc. in the filtrate are reabsorbed actively, whereas the nitrogenous waste are reabsorbed by the passive transport. Reabsorption of water also occurs passively in the initial segment of the nephron. During urine formation, the tubular cells secrete substances like H+, K+, and ammonia into the filtrate. The tubular secretion is also important steps in urine formation as to helps in the maintenance of the ionic and acid base balance of the body fluids. Functions of the tubules. PCT. PCT is lined by simple cuboidal brush border epithelium which increase the surface area for reabsorption. Nearly all the essential nutrients and 70 to 80% of the electrolytes and waters are reabsorbed by this segment. PCT also helps to maintain the pH and the ionic balance of the body fluid. By selective secretion of hydrogen ion and ammonium into the filtrate and by absorption of the HCO3 from it. Henle's loop. Reabsorption is minimum in its ascending limb. However, this region plays a significant role in the maintenance of high osmolarity of medullary interstitial fluid. The descending limb of loop of Henle is permeable to water but almost impermeable to electrolytes. This concentrates the filtrate as it moves down. The ascending limb is impermeable to water but allows transport of electrolytes actively or passively. Therefore, as the concentrated filtrate passes upward, it gets diluted due to the passage of electrolytes to the medullary fluids. DCT. Conditional reabsorption of sodium and water takes place in this segment. DCT is also capable of reabsorption of HCO3 and selective secretion of the hydrogen and potassium ions and NH3 to maintain the pH and sodium potassium balance in blood. Now, collecting duct. The long duct extends from the cortex of the kidney to the inner parts of the medulla. Large amount of water could be reabsorbed from this region to produce concentrated urine. This segment allows passage of small amounts of urea into the medullary interstitium to keep up the osmolarity. It also plays a role in maintenance of pH and ionic balance of the blood by selective secretion of H+ and K+ ions. Now, mechanism of concentration of the filtrate. Mammals have ability to produce a concentrated urine. The Henle's loop and vasa recta play a significant role in this. The flow of filtrate in the two limbs of loops of Henle opposite direction and thus forms a counter current. The flow of blood through the two limbs of vasa recta is also a counter current pattern. The proximity between the Henle's loop and vasa recta as well as the counter current in them help in maintaining and increasing osmolarity towards the inner medullary interstitium, that is from 300 milliosmolarity per liter in the cortex to about 1200 osmolarity per liter in the inner medulla. This gradient is mainly caused by sodium chloride and urea. Sodium chloride is transported by the ascending limb of Henle's loop, which is exchanged with the descending limb of vasa recta. Sodium chloride is returned to the interstitium by ascending portion of the vasa recta. Similarly, the small amount of urea enters the thin segment of the ascending limb of Henle's loop, which is transported back to the interstitium by collecting tubule. The above described transport of substances facilitated by the special arrangement of Henle's loop and vasa recta is called countercurrent mechanism. The mechanism helps to maintain a concentration gradient in the medullary interstitium. Presence of such interstitial gradient helps in easy passage of water from the collecting tubule, thereby concentrating the filtrate. Human kidney can produce urine nearly 4 times concentrated than the initial filtrate form. Regulation of kidney function. The functioning of kidney is efficiently monitored and regulated by the hormonal feedback mechanism involving the hypothalamus, JGA, and to certain extent the heart. Osmoreceptors in the body are activated by changes in the blood volume, body fluids volume, and ionic concentration. And excessive loss of fluid from the body can activate these receptors, which stimulates the hypothalamus to release antidiuretic hormone ADH or vasopressin from the neurohypophysis. ADH facilitates the water reabsorption from the latter parts of the tubule, thereby preventing diuresis. And increase in the blood volume can switch off the osmoreceptors and suppress the ADH release to complete the feedback. ADH can also affect the kidney functions by its constrictory effects on blood vessels. This causes an increase in blood pressure, and increase in blood pressure can increase the glomerular blood flow and thereby the GFR. The JGA play a complex regulatory role. A fall in glomerular blood flow, glomerular blood pressure, GFR can activate the JG cell to release renin, which converts angiotensin in blood to angiotensin 1 and vasoconstrictor, increase the glomerular blood pressure and thereby GFR. Angiotensin 2 also activates the adrenal cortex to release the aldosterone. Aldosterone causes reabsorption of sodium ion and water from the distal part of the tubule. So this also leads to an increase in blood pressure and GFR. This complex mechanism is generally known as the renin-angiotensin mechanism. An increase in blood to the atria of the heart can cause the release of atrial natriuretic factor ANF. ANF can cause vasodilation, dilation of the blood vessels, and thereby decrease the blood pressure. ANF mechanism therefore act as check on the renin-angiotensin mechanism. Micturition. Urine formed by the nephron is ultimately carried to the urinary bladder, where it is stored until a voluntary signal is given to the central nervous system. This signal is initiated by the stretching of urinary bladder and it gets filled with the urine. In response, the stretch receptor on the walls of the bladder send signal to the CNS. The CNS passes on motor message to initiate the concentration of smooth muscles of the bladder and a simultaneous relaxation of the urethral sphincter, causing the release of the urine. The process of release of the urine is called micturition, and the neural mechanism causing it is called micturition reflex. An adult human excretes on an average 1 to 1.5 liter of urine per day. The urine formed is a light yellow colored watery fluid which is slightly acidic, pH 6.0, and has a characteristic odor. On an average, 25 to 30 gram of urea is excreted out per day. Vigorous condition can affect the characteristic of urine. Analysis of urine helps in clinical diagnosis of many metabolic disorders as well as malfunctioning of the kidney. For example, presence of glucose, glycosuria, and ketone bodies ketonuria in urine are indicative of diabetes mellitus. Role of other organs in excretion. Other than the kidney, lungs, liver, skin also helps in elimination of the excretivate. Our lungs remove large amount of carbon dioxide, approximately 200 ml per minute, and also significant quantities of water every day. Liver, the largest gland in our body, secretes bile-containing substance like bilirubin, and cholesterol, degraded steroid hormone, vitamins, and drugs. Most of these substances ultimately passes out along with the digestive waste. The sweat and sebaceous gland in the skin can eliminate certain substances through their secretion. Sweat produced by the sweat glands and is a watery fluid containing sodium chloride, amounts of urea, lactic acid, etc. Though the primary function of the sweat is to facilitate the cooling effect of the body surface, it also helps in removal of some of the waste mentioned above. Sebaceous gland eliminates certain substances like sterols, hydrocarbons, waxes through the sebum. This secretion provides a protective oily covering for the skin. Do you know that the small amount of nitrogenous waste could be eliminated through the saliva too? Disorders of the excretory system. Malfunctioning of the kidney can lead to the accumulation of urea in blood, a condition called uremia, which is slightly harmful and may lead to the kidney failure. In such patients, urea can be removed by the process called hemodialysis. During the process of hemodialysis, the blood drained from the convenient artery is pumped into the dialysing unit called artificial kidney. Blood drained from a convenient artery is pumped into a dialysing unit after adding an anticoagulant like heparin. The unit contains a coiled cellophane tube surrounded by a fluid having the same composition as that of the plasma except the nitrogenous waste. The porous cellophane membrane of the tubule allows the passage of molecules based on the concentration gradient. As nitrogenous waste are absent in the dialysing fluid, these substances freely move out, thereby clearing the blood. The clear blood is pumped back into the body through a vein after adding antiheparin to it. This method is boon for the thousands of the uremic patients all over the world. Kidney transplantation is the ultimate method in correction of the acute renal failure, kidney failure. A functioning kidney is used in the transplantation from a donor, preferably a close relative, to minimize its chances of rejection by the immune system of the host. Modern clinical procedures have increased the success rate of such a complicated technique. Renal calculi, a stone or insoluble mass of crystallized salts, oxalates, etc. formed within the kidney. Glomerulonephritis, inflammation of glomeruli of kidney. So that's it. Thank you so much.